Good morning, everybody. Welcome to this HIV Center Grand Rounds. My name is Theo Sanford. I'm the director of the Development Core. The director of the center is at a conference, cannot be here this morning, so you have to do it with me today. Welcome. We have a very special speaker who I will introduce in a few minutes. Before doing that, I want to share a few announcements with you. Upcoming grand rounds on March 9th, we have two of our postdoc alumni presenting. The first one will be Omar Martinez. He will be talking about sustainable solutions for continuity of HIV care through medical legal partnerships. That's going to be very exciting because it's a new perspective that he brings to our HIV research. And the second speaker, the second alumni postdoc is Ruben Robbins, who will be talking about cognitive testing and making it equitable. Then on March 23rd, we have a very special grand rounds. There will be a presentation about a project that AIDS Institute in New York State and several partners in New York City that are doing together to improve the quality of healthcare for aging persons living with HIV. And it's a project in which consumers, what they call consumers, persons living with HIV, play a very important role. It's going to be very interesting to hear about that. On April 6th, we have early stage investigators presenting. Marina Kotze from South Africa, who just defended her doctoral thesis about the role of research in HIV, in behavioral HIV research. And the second speaker will be Brian Kuttner, who will be talking about his research that he's doing about stigma and anal sex. And then on April 20th, and that's the last one I will mention, we have Qureshi Abdul Karim presenting on doing HIV research during a pandemic. And she will talk about experiences and lessons learned from South Africa. Those are the upcoming rounds. If anybody else would like to make an announcement, please let me know. And if not, then I'm going to introduce our speaker for today. Our speaker for today is Dr. Adam Carrico. He is a clinical psychologist. At this moment, he is a tenured professor in the Department of Public Health Sciences at the Miller School of Medicine at the University of Miami. But he just informed me that he is moving to another post. He is going to be the chair of the Health Promotion and Disease Prevention Department at Florida International University in the summer. And that's going to be quite a responsibility, as if he doesn't have enough responsibilities already. Dr. Carrico works in the interdisciplinary field of psychoneuroimmunology, and psychoneuroimmunology examines the bidirectional. Wow, I'm struggling with the words, Adam, and I think out of your mouth they will come much more easy.
We call it PNI for short, so it makes it easy.
Good. So PNI examines the bidirectional connections between the central nervous system and the immune system. And the focus of Dr. Carrico's clinical and biobehavioral research program is on developing comprehensive approaches to address a resurgent epidemic of methamphetamine use. And methamphetamine use is undermining the benefits of biomedical approaches to HIV prevention in sexual minority men. So that's an important topic to address. I had a chance to review Dr. Carrico's CV, and it demonstrates his all-round competence in research, training, and mentoring, and in directing programs. He is the PI on many NIH-funded grants, and he is also co-investigator on other grants, including the Max Wise Combined Cohort Study. He's a developmental core director of the Miami Center for AIDS Research, and he has an extensive list of publications in high impact journals. He won prestigious awards, and last but not least, he is an inspiring mentor for the next generation of HIV researchers. Dr. Carrico, welcome to this Grand Rounds, and we're looking forward to your talk.
Thank you. That was very nice. So let me go ahead and share my slides here. All right. So, today I'll be talking about bridging the divide, optimizing biomedical HIV prevention from cytokines to society. And I chose this topic because it really encapsulates my experience as a predominantly behavioral scientist trying to connect the dots and, some of these walls that have been put up arbitrarily between us and our biomedically focused colleagues. Of course, that has really changed in the era of treatment as prevention and PrEP. And I think there are additional exciting opportunities to fully integrate these agendas that I'll describe a bit throughout the talk. So just an overview of what I'll be discussing. I'll review some of our work in stimulant use specifically, which has been my primary focus, methamphetamine predominantly, but also co-use often of powder cocaine or crack cocaine in the men that we serve. I'll look at the relevance of stimulant use for HIV treatment as prevention, biomarkers relevant to HIV pathogenesis, and relevance to HIV acquisition, so primary prevention of HIV. And then I'll end with some conclusions that highlight future directions. So, before beginning, I wanted to introduce some of our current conceptual models. Although our data on this are just emerging, I think it's really important to think about these multi-level determinants. A lot of my work in P&I has really been in this linkage of the exposome, we would call it. So thinking about adversity across the life course, substance use, minority stress, many of these exposures that our populations have or experience, and how that relates to many of these biomarkers, including viral suppression. But, increasingly, I think many of us are realizing that these behaviors and stressors exist in profound social and structural context. So we've really kind of integrated with Dustin Duncan, as well as Vanessa Williams, who's a K23 applicant, some of Dustin's understanding of multi-level influences, including neighborhood characteristics, but also structural and social factors, and how those could directly impact the exposome, but also moderate its associations, right? So maybe substance use matters for people that live in more disorganized neighborhoods, or for people that are homeless. So an example of moderation. But we also hypothesize that many of these kind of structural, social, and neighborhood components could directly impact HIV pathogenesis. So this nesting of individuals within their environments, we think is an important and provocative model to continue to unify the fields. And one thing that I really enjoy about it is that it's destigmatizing. We often view substance use as societally a disease of choice or a moral failure. And I think we've done a lot as a addiction research community to say that addiction is a brain disease. But it's also a social disease. We know that addiction is catalyzed and amplified by the environment and developmental exposures and stressors, including sexual minority stress. So taking this multi-level approach, we hope, helps people de-emphasize the primacy of substance use and other individual level exposures. So what I'll do is I'll take you through kind of a brief tour of what we've been doing over the years. And one thing that I always like to communicate to folks is that the meth use epidemic has been going on for at least 20 years. There is a sense that it has recently resurged, But I think it's been driving HIV treatment as prevention outcomes and HIV pathogenesis during this period. The first group to really report this association of meth with viral load was the UCSD group led by Dr. Ellis. And you see here that these are individuals that were on HAART versus not on HAART. And there was an association of meth use, those that were tox positive in their urine with both former users as well as never users, right? So it was really about the active use that was associated with elevated viral load. And for a long time, we were really interested in, are there biological direct effects of methamphetamine on HIV replication? We explored some of this in the Healthy Living Project, and you see that the answer to this is similar to the Ellis paper, where if there were effects on HIV replication, we would see them in those that were not treated. But we do really see the associations of stimulant use. These are regular users, so I think 2 to 3 times a month or more in this study, where those with the dashed bar had significantly higher viral load than their peers on ART that were not regular users. But interestingly, they do derive benefit from ART, right? You see this reduction in their viral load. So they're still benefiting, albeit a little less than their peers that weren't using. So we were curious as to how much of this is due to adherence. This is a path model where we were interested in affect regulation as a primary predictor. So this ability to reduce negative affect while enhancing positive states of mind relevant to positive affect. And all of these paths were statistically significant. So we found that affect regulation was a driver of decreased stimulant use and lower ART adherence. Stimulant users were more likely to be non-adherent, and that of course explained viral load. But the interesting thing for us is there was this residual association, this is all baseline data, of stimulant use with viral load. And so we're really curious about what explains that. Are there biobehavioral mechanisms relevant to that? We looked at this in longitudinal data with the NIMH Healthy Living study, which was the baseline data from before. And we really found that depressive symptoms and weekly stimulant use or more predicted a higher mean viral load over that 25 months. Interestingly, there were separate mechanisms relevant to ART persistence that explain this. Those with elevated depressive symptoms were more likely to stop their ART, and then that explained their mean viral load over that 25 months. And the weekly stimulant users were more likely to go on and off ART, explaining their elevated mean viral load over that period. And this is provocative because stimulant users are essentially doing the SMART trial to themselves, where we thought that for a period of HIV research, stopping medications could be good for patients to give them a break from some of the more toxicities related to legacy regimens. And it leads to resistance, it leads to elevated inflammation, and just bad outcomes overall. So this kind of intermittent pattern of utilization, I think, is a really important target for stimulant users to kind of keep them in HIV care. So that was then, right? Like we know about the older era, and I just wanted everyone to know as well that things have changed so dramatically. And I think we don't emphasize this enough. But right around 2012, we got medications, integrase strand transfer inhibitors that are much more forgiving of non-adherence, where people can achieve viral suppression even at 70 or 75% of their medications taken. And so data on the left here are from the CNIX cohorts, where you see that there's been a steady linear progression of viral suppression over time, such that by 2015, 90% of the people that were in HIV care in these clinics were virally suppressed. So things have gotten tremendously better, and we have much better tools available to help people manage HIV infection with minimal side effects and more forgiving of non-adherence. You see data on the right here from the SCOPE cohort at San Francisco General Hospital, where we wanted to look at this time-varying association of stimulant use with probability of viral suppression. And you see in the earlier years of SCOPE, there was this difference, right, where about 70% or so of those that used every single visit were virally suppressed, versus those that used it half their visits versus those that used it none. The magnitude of this difference has really shrunk over time, such that even stimulant users have crossed that 90% threshold in San Francisco at Zuckerberg San Francisco General Hospital, right? So this is not the real world, as they would say, but it is part of what we can all aspire to, because San Francisco is such a special place. So this is some of our data on sexual minority men who use methamphetamine that were living with HIV. My first R01 was really focused on viral suppression as the primary outcome. And we were really surprised early on in 2013 to see that men were coming in and for our baseline assessments, and many of them were undetectable. And about 2/3 had undetectable viral load, and even more were virally suppressed, less than 200 copies. So you see the HIV care continuum here that we published in this paper with Matthew Mimiaga and others, where most of our guys were in care, a few more were on ART, right? So they were getting their prescriptions, just not attending HIV care. 2/3 had an undetectable viral load. But the real problem here is when you look at the percent that reported they were adherent to their ART, 90% of doses or more. So that was only 30%. And only 1 in 4 had both, right? So they had undetectable viral load and adherent to ART. And we know that's so important, not just to remain virally suppressed, but adherence, even in those with undetectable viral load, can reduce inflammation and other immune markers relevant to health outcomes in people with HIV. And also the signal to us earlier on that people were at high risk for virologic rebound. We know that it's not just about getting virally suppressed or undetectable, but staying that way. Going along the conceptual model that I presented at the beginning of the talk, we also have examined some of these social determinants of these HIV care cascade outcomes in this population and found that homelessness was associated with 80% lower odds of this composite outcome, undetectable viral load and adherent. And being in substance use disorder treatment, they had 3-fold greater likelihood or odds rather of being adherent to ART. So some indication here that it's not just the drugs, right? That there are these structural contexts and the availability of substance use treatment that could optimize HIV treatment as prevention in this group. So I'll present some of the data throughout this talk on Artemis. It was my first R01. It was a randomized controlled trial testing a positive affect intervention that I'll talk a little bit about. But the kind of platform for Artemis was contingency management. And CM, for those who don't know it, also is called conditional cash transfer, when you want to destigmatize it and make it not about drug users, or financial incentives. There are lots of ways that we've talked about the same thing in different literatures. But CM is a evidence-based approach where individuals receive positive reinforcement for behavior change, often toxicologically negative stimulant urines, so negative for methamphetamine and cocaine metabolites. And you see here that this meta-analysis from Prendergast, the magnitude of those effects of contingency management are moderate, about 0.66 Cohen's d. But the problem is what happens after the incentives end. Many trials have shown that CM, or a few trials have shown that CM can reduce viral load in the short term, but not after the incentives are discontinued. And so the premise of Artemis was, can we deliver a positive affect intervention during CM to help people boost and extend its benefits with regard to viral suppression? And why positive affect? I tell people it's not unicorns and rainbows and sunshine and kittens. It has a underlying neurobehavioral basis. And the reasons that we have chosen to focus on positive affect are twofold. One is just withdrawal. when individuals go through contingency management, stopping methamphetamine use produces predictable increases in depression and other forms of negative affect. Individuals can receive these positive affect skills to help them cope or manage those withdrawal symptoms and hopefully get the most out of their time in contingency management. The other, I think, more provocative neurobehavioral process is what we call hyporesponsivity to reward, or anhedonia. I think this is a common symptom in many psychiatric disorders, and people don't often associate it with substance use disorders, but there are well-characterized deficits in reward processing of natural rewards, even primary reinforcers like food, money, sex, where the dopamine reward system is just less active in people with substance use disorders. So the idea of a positive affect intervention was to sensitize individuals to these natural sources of reward and reinforcement to kind of change that pair mix of their dopamine reward system. So we think that delivering that positive affect intervention during evidence-based substance use approach like contingency management could yield better long-term outcomes. And this positive affect work stems from Judy Moskowitz. She conducted RCT of the intervention in people newly diagnosed with HIV and found that it decreased HIV-specific distress and reduced antidepressant use. So some good premise for the ability of the intervention to impact these sorts of mental health outcomes. And what do we find in the Artemis trial? Artemis stands for Affect Regulation Treatment to Enhance Meth Intervention Success. And so everyone received the contingency management. Some people got 5 individually delivered Artemis sessions, which include, positive reappraisal skills, formal informal mindfulness, noting positive events, gratitude journaling, a variety of different skills that are all focused around increasing positive emotion. And the attention controls completed self-report measures and neutral writing exercises. So they were matched for time, attention, and incentive. And we enrolled 110 sexual minority men with HIV that had biologically confirmed meth use. So these men tested positive in either urine or hair, and that helped us really establish that they were eligible and not just trying to receive all of these CM incentives. And at baseline, as I mentioned, we had a high prevalence of viral suppression, such that only 14% of participants were unsuppressed. Their viral load was greater than 200. And you see the story of Artemis here is that we help people stay steady at that baseline level. 15% of our guys had at least one unsuppressed viral load over the 15-month follow-up in Artemis. And that's compared to, so I have to move them, 46% of our guys in the attention control. So there was this effect on virologic rebound, really highlighting that people need help staying suppressed. We've also looked at some of these mechanisms. There were concurrent increases in positive affect through 12 months, as well as decreases in self-reported stimulant use through 12 months. And one thing that we find, this is Jiyoung Lee on my team, who was a pre-doc, that these long-term reductions in stimulant use were associated with viral load at 15 months. They predicted it. Not so much for the positive affect changes, right? So it does seem to be that the intervention effects stem from these self-report reductions in stimulant use, which is nice because many people look at self-reported substance use and think, what does it mean anyway? And we have some predictive validity here that it's linked to viral load at the final 15-month time point, adjusting for baseline. Interestingly, there was some residual effect of the intervention even after we adjusted for stimulant use that couldn't be explained by these positive affect changes. So I think more work to do to unravel those mediators. Another random interesting fact that emerged in this study is that elevated positive affect at baseline predicted better or lower stimulant use after the 3 months of contingency management. So it could be a good prognostic indicator for those entering treatment. We've moved on in Artemis to look at a variety of biological outcomes that I'll discuss. And this is data from a subset of 53 participants who had sustained virologic control. So they were undetectable at baseline and at that 6-month follow-up that we are looking at here. And this is so important because we know that viral replication impacts a variety of cellular and extracellular processes, including DNA methylation, which we examined here. We conducted epic arrays. So we saved the DNA from everybody in the trial at each of their visits when we drew blood. And then we looked at methylation. And these are CpG groups that get added on top of DNA or removed from DNA. Almost think of like a curtain that covers or opens a flashlight or a stage light. And so that is a way that cellular processes are regulated. When something is methylated, it's not being coded for by mRNA and proteins and all of these cellular processes. When it's demethylated, it is coding. And so what we looked at was whether or not Artemis, the positive affect intervention, decreased very specific methylation sites for some of the neuroimmune signaling mechanisms that we thought would be relevant to immunologic outcomes. So some of the glucocorticoid as well as adrenergic effects, we thought it would reduce stress hormones like norepinephrine and cortisol. We also thought the positive affect intervention could amplify or heighten compensatory processes neurobehaviorally, like oxytocin, which is the love hormone, for those who don't know, and then increase BDNF, which is thought to be a neuroprotective hormone or neurotrophin. And so we looked at the receptor level on the cells for the most part, these beta-2 adrenergic receptors, glucocorticoid, oxytocin receptors, and BDNF promoters. What we find is somewhat of a mixed bag in terms of methylation and demethylation. We're still sorting through these results, but we do see effects of the intervention on reducing beta-2 adrenergic receptors. And so that is very exciting for us because those are the primary receptors for norepinephrine, which has been linked to a variety of negative outcomes in people with HIV. And we see decreases in oxytocin receptors. So, this tells us that these are, coming back to normal, right? We're decreasing that methylation. The cells are able to receive more input from those because meth is not driving the dysfunction. We've also been looking at different pathways. We know that genes and methylation sites on genes function in families. And so we've been looking at how they work together to impact immune function and neural function, as well as measuring some of these concomitant changes in soluble markers for immune activation and inflammation that I won't chat about today. But just the pathways, here's what we see with them. They're kind of mind-bending and beautiful. We see significant intervention-related improvements in neurogenesis and cellular signaling using our GO pathways. And these are what we call FDR adjusted, false discovery rate adjusted p-values less than 0.05. And so we're taking into account the multitude of tests that we have to do to run these pathway analyses. I think more exciting for us, we looked at the reactome pathways. We saw intervention alterations in 75 of them at the FDR adjusted p of 0.05. And many of our pathways are showing improvements in neural functioning like synaptic transmission, but also improvements in dopamine, glutamate, and serotonin neurotransmitter release. So, at a cellular level, we are seeing an epigenetic methylation signature for an effective positive affect intervention or effective behavioral intervention for meth. So at the end of the day, we can see a neuroimmune profile relevant to recovery, which is very exciting. And next, I'll talk a bit about pathogenesis and some of the work that we've been doing with Artemis samples and other data sets to really understand this kind of gut immune dysfunction that is really the hallmark of HIV. We know HIV damages the gut very early in infection, leading to persistent leakage of microbial products like lipopolysaccharide and other microbes from the gut that when they make it into the periphery are recognized as foreign and trigger the immune response with immune activation and inflammation. A lot of our work is focused on amino acid precursors for neurotransmitters like tryptophan. And so this chronically overactive immune system in HIV or other chronic viral infections depletes tryptophan. So it robs people of the building blocks for serotonin, and it has well documented neurobehavioral sequelae that could in turn increase risk for meth use, right? So when we overlay meth on this, we know meth amplifies that gut damage. That's a lot of what the data I'll show you, as well as this gut immune dysfunction and the pathway. So, we think there are these bidirectional connections of methamphetamine and the brain through this gut-brain axis. Just a little bit of our data. We've done RNA sequencing, so this time looking at messenger RNA with 55 guys from Artemis that were virally suppressed. So again, less than 200 copies per ml. In this study, we looked at whether or not they were tox positive in urine. So when they had that blood draw, did they have a sample that was reactive for often methamphetamine, but also at times cocaine? And we saw 32 differentially expressed single genes in those that had a tox positive urine sample, including genes relevant to HIV latency, which was exciting, and immune activation and cell cycle regulation. So it could be that meth use is impacting the reservoir at a cellular level from some of these data. And we also saw concomitant changes in pathways. So this is two-directional perturbation of pathways relevant to a variety of immune functions, like you see chemokine signaling, T cell receptor signaling, and so on, but also many of these amino acid precursors relevant to serotonin, as well as catecholamines, including dopamine, with the tyrosine metabolism. We also have looked in the plasma to see what's floating around in guys' blood. This time we had 84 men that were undetectable. Again, those that had a tox positive urine at that blood draw showed a significantly elevated soluble CD14. And SCD14 is exciting because it's a monocyte activation marker that's really secondary to that translocation of microbial products from the gut. So it gives us some indication that meth or other stimulants could be making the gut leak due to damage or changes in autonomic function. And that could have implications for activation of this monocyte marker. Coincidentally, SCD14 predicts faster clinical HIV progression, even after we adjust for viral load and CD4. So it is very relevant to the health of people living with HIV. More recently, we have conducted a COVID-19 supplement, and this is work being done by Emily Sharenak in our lab. She's a postdoc with her F32 funding. And so what we have done here, we enrolled 75 men who completed an online survey and then came in to provide biospecimens for SARS-CoV-2 testing and blood draws to measure their plasma, sera, and peripheral blood mononuclear cells, or PBMCs. And what Emily finds in these data is that controlling for HIV status, stimulant use is associated with higher SCD14, even in those that are HIV negative. And you see over here that the association is actually most pronounced in those that are HIV negative compared to the positives in orange. That interaction is not significant, largely due to power in this smaller study. But I think that we can say that there are independent associations of stimulant use on this monocyte activation and even in negative men, so not confounded with HIV pathogenesis and the damage that HIV has done to the gut, or at least not amplified by that. There were also concomitant elevations in C-reactive protein at a level of 1 or greater. And CRP is just a marker of systemic inflammation that's released from the liver after it's stimulated by IL-6. Interestingly, we saw no associations of stimulants with SARS-CoV-2 infection measured using RNA-positive nasal swabs, but also nucleocapsid antibodies. So these are antibodies to other aspects of CoV-2 that are not impacted by vaccination, like the spike protein antibodies. And so we've moved on again in the social direction to look at social determinants of soluble CD14. This is work being done by Dalaram Ganuni in our lab, who's a PhD student, and she is finding associations of being unemployed, arrested, which is also a proxy for severity of stimulant use, homeless. These are all associated with different measures of gut immune dysfunction in our studies. And you see on the right here just these associations with soluble CD14 in the orange box plots. Those that were arrested and those that were homeless in the past year had elevated soluble CD14. And this is coincidentally even after adjusting for HIV status and stimulant use. So there is something about these structural determinants over and above substance use, which is exciting and I think important to see. And again, it's not just the drugs. I think I keep hammering that home, which I'm happy for. But many of these behaviors like stimulant use and depression and negative affect exist in a social context and are stemming from the social stigma, often an intersectional stigma that our men experience and other high priority populations experience. So this is the minority stress model, which I'm sure folks know, that there are these kind of distinct dimensions of minority stress, like prejudice and discrimination, anticipating stigma, internalized stigma or heterosexism in this instance, and concealment or not being out about your identity. And so what we hypothesized is that in the context of treated HIV, these sexual minority stress processes would potentiate stimulant use or more frequent stimulant use and greater negative affect, which would dysregulate the autonomic nervous system and the hypothalamic pituitary adrenal axis. And these hormonal influences hit the cell, right? There are receptors on the cell that impact the transcriptional regulation of the genome, essentially. And so DNA methylation, which we discussed earlier, but once things become methylated, they kind of open up and transcribe into mRNA. So in this next study, we're looking at methylation again, but it's nice to know this transcriptional pathway that really underlies some of the cellular changes that we see in HIV. As we age, period, things like immunosenescence, so more exhausted or fewer naive T cells, but also shorter telomere length, which are the shoelace caps for your chromosomes. So you see them kind of here, they protect it. And in this study with, again, Dalaram and Anissa Felengi, we looked at 52 meth-using or sexual minority men that were undetectable, and we used the EPIC array again. And this time we looked at composite measures from these EPIC arrays that index accelerated biological aging, so a faster epigenetic clock. So my chronological age is 44, and hopefully my biological age is 34, right? But it could also work in the other direction. And so a higher epigenetic clock is indicative of accelerated biological aging. We looked at telomere length, naive CD4 and CD8 T cells that were predicted from these EPIC arrays. And of course, we want all those to be higher in people with HIV and without HIV. So these are our bivariate associations of a composite measure of sexual minority stress with a faster epigenetic clock, with shorter telomere length, with fewer naive CD4 and CD8 T cells. There was this robust signal of sexual minority stress. And for the types of work that we do, these correlations are quite large in magnitude, albeit in a small sample. So please take them with a grain of salt. And what we did was we adjusted for prevailing levels of negative affect and recent stimulant use to see, does this still hold? Is it still there? Is that association for sexual minority stress there? And for sure. It definitely held, and we saw those same associations of the composite sexual minority stress measure. But also we found that greater outness was associated with more naive CD4 and CD8 T cells and lower plasma inflammation, lower IL-6. So I like the take home of this paper that stress is bad, as many of us know, but being out has benefits for men's health. And lastly, I'll kind of move along here. I know we're running a little short, just on comprehensive approaches to HIV prevention in people who use substances. And, I would say that this literature is relatively nascent with regard to interventions along the PrEP care continuum, where many trials are underway, including some by our group, to optimize PrEP uptake, PrEP use, PrEP adherence, and persistence. And, in general, I think the approaches are integrative in nature with mobile access units that are being done in HPTN, delivering kind of comprehensive services like substance use treatment, mental health, or HCV treatment, and adapting many of these evidence-based behavioral interventions like the Artemis intervention or motivational interviewing and so on to target the PrEP cascade or continuum indicators. And when we look at people who inject drugs, the PrEP literature, I think, is relatively focused on awareness, right? But also willingness. There's not been as much work on actual PrEP use or adherence in this group. And there really is a problem with PrEP uptake. We see low uptake across studies of people who inject drugs. And so there is really a need to optimize that uptake and get into needle exchanges, mobile units, and other work that folks outside of my specific area are doing. We also looked at the PrEP care continuum in substance-using sexual minority men. And this was an interesting contrast where, we find that sexual minority men that use substances are as likely or more likely to be taking PrEP. But those that use stimulants such as meth have lower adherence to daily oral medications, right? So the problem is not uptake, it's actual adherence when they're taking these medications, and lower persistence is seen as well. So the problem is really at a later stage, or the barriers that this population experiences are a later stage of that continuum. And I would also say there is this thread in the literature that's fascinating around event-based dosing, and that it may be better adherence for men who use chemsex drugs or stimulants. So maybe different dosing strategies, maybe injectable PrEP. There are other options that we have available to optimize HIV prevention in men that use stimulants. This review was recently published, so you can kind of see all of the relevant studies. And I'll move on a bit to talk just about our work relevant to HIV prevention and some of these biological alterations in the rectal mucosa. Surprisingly, mucosal immunology in sexual minority men has not been as robust, despite it accounting for 70% of all HIV infections in sexual minority men, receptive anal sex. So really understanding what's going on in the rectal mucosa is essential to ending the epidemic, to identifying viable targets for new pharmacologic approaches, including microbicides in like douches and things that can, actively address some of these underlying biological alterations that we see secondary to receptive anal intercourse. And of course, meth use has continued to be linked to 1 in 3 new HIV infections. These are recent data from Christian Grove's T5K cohort, and that number has actually increased over time, where the lion's share of infections that we're seeing are in men that use meth. And so what we were interested in here is trying to understand biobehavioral risk factors that could amplify biological vulnerability to HIV by changing rectal immune function. We know that meth and other stimulants make people more impulsive. They have more sex for longer periods with multiple partners. And of course, there are these behavioral pathways. But what we're interested in specifically is, are these psychosocial and behavioral factors linked to amplified dysregulation of the rectal immune environment? And we do see some evidence for that from our studies and others. On the right here, this was led by Greg Tapia and Tiffany Glenn. You see our red dots are those with 2 or more syndemic conditions. So stimulant use, hazardous alcohol use, PTSD, and depression. So those with 2 or more of those conditions had elevations in several rectal inflammatory cytokines or chemokines. This is about 68 men that were recruited in STI clinics in Miami. And these elevations in rectal cytokines and chemokines were relevant not only to inflammation and immune activation, but actually like recruitment of Th17 cells, which are the primary targets for HIV infection. So some really fascinating data here that on a biological level, these syndemic conditions could be amplifying vulnerability to HIV. Of course, PrEP can mitigate all that, right? So getting people on PrEP is still really important, but understanding some of these biological processes could guide new approaches. We're currently conducting Amethyst, the American Transformative HIV Study, led by Christian Grove and myself, to really look at methamphetamine and its association with HIV incidence and see how that is modified by a variety of structural factors, which Dustin will be measuring in that study, but also looking at rectal cytokines and chemokines to understand if meth use amplifies biological vulnerability. And it's also about the microbiome and the rectal microbiome. So I just wanted to put a plug in here for Angela and Jen Manusak, who are looking at our microbiome data from that prior study. And so we did 16S sequencing of the gut microbiota to really understand, are there differences as a function of behavioral factors? In this instance, we looked at douching because it seemed to be probably the most mechanistically linked to the microbiome. And we find that those who douche in the red box plots here have lower richness, evenness, and Shannon diversity for their gut microbiota. So these are all considered to be bad things. We want a more diverse gut microbiome. There were also concomitant perturbations in the cytokines and the cytokines and chemokines that I was talking about before. So, a lot of data here that points to understanding exactly how and why these psychosocial and behavioral factors impact rectal immune function, and really points to the need for, I think, douches that would have tenofovir diphosphate or other preventative compounds for HIV acquisition. Not everyone is going to want to take PrEP. Not everyone is going to want to get an injection. And so this could be another viable pathway that's actively being explored within the ATN by Jose Bauermeister and others. These are actually the gut bacteria, which there wasn't as consistent of a sign, but you see how much data we get on different bacterial species. So it's a pretty exciting world to do this microbiome work. And just in conclusion, I think I've taken you kind of on a tour of the work that we've done in stimulant use, and I hope that you're walking away with an appreciation of our need to really optimize the benefits of treatment as prevention and PrEP in those who use stimulants, and that we need comprehensive biobehavioral approaches, including behavioral interventions, but also pharmacotherapies that are being tested for methamphetamine use and other stimulant use disorders. And these treatments can not only improve viral suppression or PrEP adherence, but they could also address neuroimmune dysregulation that we see, especially in people with HIV who use stimulants. And so our Artemis data are really some of the first data that effective meth treatment could modify epigenetic signaling and have an epigenetic signature. And of course, behavioral factors can heighten HIV acquisition at a biological level via rectal immune dysregulation. And as I've been saying throughout, we really feel like an important next step is understanding these individual level exposures in the context of multi-level determinants, not only to guide comprehensive care approaches, but reduce the stigma that people who use stimulants experience. So that's it.
Adam, thank you so much. This was an incredible overview of a very comprehensive research program with a variety of HIV-related outcomes, with multi-level determinants, incredibly impressive. And what impresses me is all these things come together and hang together. And I like it very much that you started with a model. That model came back several times, and it really integrates the findings of these all these different strains of this comprehensive program. Very, very impressive. So congratulations, and thank you for sharing that with us. Let's see if there are any questions. If you have a question, raise your hand or put something in the chat. And this is the opportunity to ask Dr. Carrico questions that you are interested in. And I know, Adam, that in our audience, there are people who are familiar with this kind of approaches and this kind of work. So I assume that they will have questions for you.
Nice. And I guess I would say one person I did not speak about was Brad Awizarot. He's there at NYU in New York, and he has been a lovely mentor and colleague and is a genomics expert. So, doing this kind of work, I would say don't be afraid to cross the boundaries and find good people that can actually accomplish it. It takes a village to get many of these omics studies done.
And what I understand, Adam, based on what you're just saying now, is that your work is a continuing discovery process where you expand beyond your own discipline and integrate much more knowledge from other disciplines, and that must be an amazing journey.
It's kind of like having a library card. I think I don't know. So sometimes I'm a mile wide and an inch thick, and you just have to acknowledge that. But Susan has a great question on wondering the role of psychotropic medications. I think that's just critical, right? And that we are in great need of pharmacotherapies for methamphetamine use disorder in particular. There are preliminary signals that are quite positive for mirtazapine, which is an atypical antipsychotic, or maybe antidepressant, and also naltrexone with bupropion. And so, I think those trials are going on, but meth is such a complex problem that also occurs in a social context. And so I think testing these multi-level interventions, it's going to take the package, right? Like we talk about the HIV prevention toolkit, and we'll need something similar for people who use stimulants. It's not just going to be one thing. But I think the more that we can interact with people doing those pharmacotherapy trials, I think it'll be better for the fields.
Susan, that answers your question? Let's hope it did. There are two participants who raised their hand. Okay, let's start with Ruben. Ruben Robbins, you want to come into the room and ask your question?
Hey, Ruben.
Hi, Adam. Can you hear me?
Yeah.
Okay, great. Thanks. Great to see you and great talk. And as Teo mentioned, it's a really impressive body of work. I don't have a specific question as much as I'm just curious to hear your thoughts on the complexities of understanding how all of, I'll say all of life can affect the immune system and health outcomes, and how we how can we best begin to parse out all the different types of exposures, whether it's to meth or HIV or homelessness or being arrested or living in poverty, being exposed to chronic air pollution or other infectious diseases. All of those can affect our immune system and actually affect health outcomes. There's so many different exposures. How do we best parse all this out to understand whether it's in HIV or other diseases, how this affects our outcomes?
Yeah, it's kind of like, what's the attributable risk of each of these predictors? And I think it's a great unanswered question in many ways, right? And so I think the first step is to conceptually acknowledge that complexity and then start to develop the models that can understand it with this nesting in the environment. I guess for myself being an interventionist, I view a clinical trial as an experiment, right? And that is the beauty of our meth treatment studies, is that we can experimentally change meth use and see this concomitant causal effect on viral load and other biological outcomes that we examine. And so, I think that top of the causal inference prism being a trial, thinking through, what it would take, right, to do a community level or a multi-level intervention targeting many of these exposures, and how much could we actually change viral suppression and other biological outcomes. So, not so much an answer, but I think the more that we can move towards multi-level interventions, I think we'll have a bigger impact, right? At the end of the day, I'm not so concerned as like how much does this and that contribute, but like, can we move the dial on meth use? Because right now, we throw our best interventions at it and they move it by 10%. So the recent Trivedi trial in NEJM for pharmacotherapy with naltrexone with bupropion, it's making a dent in meth, but it is not hitting it out of the park.
Thanks.
Yeah.
Justin Knox. Justin has a question. Go ahead, Justin.
Hey, can you hear me?
Yeah, how's it going?
Good, good. How are you, Adam?
Good.
Great presentation. As you note, though, it's a bit narrow, right? I'm wondering how you can expand your research. Just kidding, obviously, but how you have or are considering expanding your research to include implementation science and like policy work. I imagine you have thoughts around that.
Yeah, maybe. I'll see how much yeah, I'm kind of having fun in this kind of biobehavioral efficacy sphere, right? But there are definitely interventions that we're developing and testing again, like the positive affect intervention. We have trials underway in mHealth apps of it with men that are positive, as well as a telehealth, like Zoom-based therapy for guys that are on PrEP. So I feel like as that work matures a bit more, there will be these opportunities for implementation and dissemination in HIV clinical settings. The Artemis intervention was also picked up by SAMHSA in their, I think, 2IS initiatives. So it's being implemented in, I think, 2 or 3 clinics across the US for people with HIV. And so I'm supportive of all those directions. But at some point, I would have to switch from the biobehavioral work that I'm doing to be focused more on implementation. And maybe that'll happen as the arc of the interventions evolve. Yeah.
Okay, yeah, thanks. Yeah, actually, I kind of set you up here because I've been emailing with Christian about integrating some implementation science measures into your Amethyst study.
Oh, great.
Regarding men's willingness and preferences regarding, accessing treatment for meth use, which I think, this idea of integrating implementation science measures into observational research could be a way to, expand that collaboration. So maybe,
Awesome.
maybe I'll drag you into some of this work with me, but that's exciting.
That'd be fun, yeah. And I think especially the extent men are very savvy, right? Their providers are savvy. And even though, for example, the pharmacotherapies for meth use disorder are not FDA approved, many are using them off label, right? And so just documenting that use, people's attitudes and appraisals of different treatment options, I think is really important in the substance use field, because we know that only like 1 out of 7 people with a substance use disorder are actively seeking treatment. So the kinds of treatments we're providing are often unacceptable to a large proportion of the population we need to serve. And the question is, how do we develop treatments that really reach a broader population?
Right. Yeah. And to get to your point with Ruben is, can we go beyond estimating like population attributable risk and actually like population addressable risk, right? Like what could we actually do about that level of risk with the interventions and treatments that we have?
Totally.
Okay, and next question is coming from May 10, one of our postdocs, Adam. May, go ahead.
Thanks. I wasn't sure how my connection would hang out here, but I just was thinking about how, with all the complexity that seems to be localized, when you look at the nested nature of the problems, how can we figure out what's going to work locally, and then what can be more generalizable to work more globally? And then how do we develop policy based on how specific some situations might be in different, just to speak to how you mentioned San Francisco as being a very specific kind of environment.
Totally. And I think it is a challenge, right? And many of these kind of generalizability issues would happen in like a platform trial, like an HPTN or a NIDA CTN, to really, reach the broader population of men with and without HIV that use meth. We're doing a lot of that in the light cohorts. We have the Amethyst project, so we'll be able to kind of reach a national sample and do some work around PrEP uptake in that group. We're also getting a light viral suppression cohort funded that could provide a platform for doing a national trial in men with HIV that use stimulants. So I guess that's what I would say is a viable pathway to understanding whether and how the effects of these interventions vary by the structural environment, right? I mean, living in Florida, we're not a Medicaid expansion state. Our governor has his own ideas about a lot of things. I'll say only that because I'm a public employee soon. And there is this policy and structural context that impacts the effectiveness of these interventions. And in some ways, these digital health approaches are uniquely situated to answer many of those questions around generalizability and effect modification by environment.
Yeah, sounds like a big job though. Thanks.
Yeah, if someone wants to do it, you're more than welcome to join in.
So you're presenting a very comprehensive program, Adam, and then people want you to do even more.
I hope it evolves in that direction. It's like, that's the best part of what we do is I could have never expected that I would do microbiome work when I got my PhD. So, the sky's the limit. So, and you try and figure out what's the next step as you move along.
Okay, let's see if there are any other questions. I don't see any other questions, Adam. Anything you would like to say in conclusion?
No, thank you so much for the invite. I really appreciated the opportunity, and it's nice to connect and see some familiar names.
And we were very glad you were here. Yesterday, I had our annual meeting with one of our postdocs about her work plan. And this completely, what you describe is what she is interested in. So I hope she picked it up and maybe reaches out to you. So yeah, thank you so much, because this was very comprehensive and very inspiring. And I hope that will have an impact. So thank you, and thanks everybody for being here. I hope you will be able to join us on March 9th when our 2 postdoc alumni will present on their work after they left our program. Thank you and have a good rest of your day. Bye-bye.
Take care.
Bye.